Good morning to all. I am Gnanapriya, Botany Faculty of Shankar Medical Academy. So today we are going to have a detailed idea about how a chromatin reticulum changes to chromosomal structure. So I hope this is a question. I made a questionnaire and I've given you and elaborated some concepts in it. How a chromatin reticulum changes to chromosome. See, when we basically see a nucleus, the nucleus will have only chromatin reticulum. They only contain this type of a bundle of wool. This is how it clearly been present. So we call the structure as a chromatin reticulum, or we call them as chromatin fibers. So this type of chromatin reticulum or chromatin fibers are basically present in a interface of a cell. So now what do you mean by interface of the cell? Interface is called as a non-dividing cell. When a cell is not in the stage of dividing, the nucleus will show the presence of chromatin reticulum. When the cell is going to prior for a cell division, if the cell is going to get divided, that time the nucleus will show a structure of chromosomes. So that was the questionnaire which has been asked here. How a chromatin reticulum changes to chromosome. So what was the answer for this is, when a cell is in a non-dividing stage, inside the nucleus, they will be seen as a bundle of wool. We call them as what? Chromatin reticulum or a chromatin fiber. When the cell is about to divide, when they are in the process of prior division, the nucleus will show a structure of a chromosome. So this is a typical egg-shaped structure. We call this structure as a chromosome. So basically we know that the nucleus contains chromosome, the chromosome contains genes, genes contains DNA. So now we'll have a detailed idea, or we'll have a crystal clear conceptual oriented thing that whether we have chromosomes or the chromosome carries genes or the genes carry a DNA. So let me know in which sequential order the DNA molecule is present. So we must know how it happens. When it is in a non-dividing cell, that is in an interphase period, it is in the form of chromatin reticulum. When they are going to undergo a cell division, this chromatin reticulum morphologically changes to a structure called as chromosomes. So this is the real idea behind, or the justification, how a chromatin reticulum changes to a chromosome. Now, once it becomes a chromosome, what happens? We'll see the next slide, which is giving you an idea how a chromatin fiber become a chromosomes. As I drawn in the picture, you could see a bundle of wool, which we call it as chromatin fibers. These chromatin fibers, individually they contain chromatin. So chromatin fibers has got a individual units called what? Chromatin. This chromatin undergoes a process called as what? Condensation. When does the condensation occur? When the cell is going to divide, the condensation occurs. When they undergo a condensation, they create two arms, P arm, and which we call it as P arm, and this we call it as a Q arm, which is P arm represents the short arm, Q arm represents the long arm. In between the two arms, they have a centromere in the center. So this structure we call it as a chromatid. So first, how it is inside the nucleus? It is in the form of chromatin fibers. This chromatin fibers have individual units called chromatin. During the process of cell division, they undergo condensation to form a structure called as chromatid. This chromatid will undergo a process of duplication. What do you mean by duplication? The single sister chromatin will become a double sister chromatin. So now the sister chromatin becomes two. We call it as homologous sister chromatid, and they show a typical structure of X. This X-shaped structure we call it as a chromosome. So this picture will explain you how a chromatin fiber is converted to a typical structure of a chromosome. So first what you see was a bundle of wool that we call it as chromatin fiber. Each chromatin fiber unit we call it as a chromatin. During the process of cell division, they undergo condensation to form a structure called chromatid, and they form a duplication. The process of duplications, after the process of duplication, they become a homologous sister chromatids. This homologous sister chromatids are nothing but an X-shaped structure called as a chromosome. Now we got a visible structure of the chromosome. Now one more question comes. During which stage you can see the chromosome? During the phase of anaphase. During the phase of anaphase, you can see the typical structure of the chromosome. Or in this phase, you could study the different structures of the chromosomes, or the period in which you could see visibly a structure of a chromosome. So now we'll study the individual structure of a chromosome. So what is an individual structure of the chromosome? This is the individual structure. So as I said earlier, the short arm is called as P, the long arm is called as Q. In between region, we call it as a centromere. Otherwise, we call them as a primary constriction. So this is about a structure of the chromosome. So now you could see here yellow color points. You could see that as yellow color points here. We call that as the points which are present on the sides of the chromosomes are called as locus. In this locus only, we have a green color band called as a genes. So what I want to explain it again. We have an entire typical structure of the chromosomes. In the chromosomes, we have some locations or the points, we call it as locus. Every locus carries a band. That band we call it as genes. So now what do you understand from this picture? We understood that the chromosomes are the physical carriers of the genes. So now what did we understand very clearly? The chromosomes are the tool or a physical carriers of the genes. Where actually the genes are present? The genes are present on the locus of the chromosome. So what is the structures represents the locus? The yellow color points are called locus. The green color bands are called genes. Now in turn, the gene carries a macromolecule called DNA. So gene carries a macromolecule called what? DNA. So the DNA is the macromolecule. Since the genes carries the macromolecule of the DNA, genes are called as a structural functional unit of the heredity. They are called as a structural functional unit of the heredity. So the genetic information is being encoded in the genes due to the presence of the DNA. So due to the presence of a DNA. So what is a gene actually? A gene is a discrete chunk of a DNA molecule. So the genes are in the form of base pairs, and these base pairs are representing a particular genes inside of a body. When you take about the human being, we have approximately 30,000 genes. See, for example, a chromosome carries the genes carried by the DNA. So this DNA is in the form of base pairs. We have approximately 30,000 genes. So now we come in still in a better way. Still in deep, we can understand that the genes also carries a concept called as alleles. So now we come for the next concept. What is an allele? Now you know what is chromosome, where is gene is present, and where is DNA. Now I'm asking you an intermediate question. Where is the allele present? Alleles are present in the genes. What is an allele then? Allele is nothing but a variation of the gene. What is an allele? So we call it as a alternative form of a genes. What we call allele is an alternative form of a genes. So they can be either dominant or they can be either in the form of recessive. So they can carry a dominant character or they can carry a recessive character. Now the next question comes, what is dominant and what is recessive? When it is expressed, we call it as dominant. When that is not able to express itself, even if it's present there, then we call it as a recessive. So what we understand from the today's class, we said that the chromatin reticulum will become a chromosome. And the question is, how a chromatin reticulum become a chromosome? The answer is very simple. When the cell is not dividing, they look in a form of a chromatin fibers. When the cell is about to divide, or when the cell is under the process of division, or during division, the cell will contain a chromosome. Now, what was the next question come? What does actually the chromosome contain? The chromosome actually contain some certain points in the chromosomal structures. We call them as locus, and the locus is the point where they carry their genes, and this genes in turn carries the molecule called DNA. Since DNA is being carried by the genes, we call the gene as a hereditary information because it is encoded by a DNA. In simple words, the gene is a discrete chunks of a DNA molecule, and we have approximately 30,000 genes in our body. And now inappropriately, we are going to learn in a better way. The genes also contain another intermediate compound called as alleles. So now in detail, we'll see what is an allele is. So when you talk about an allele, so allele represents a particular trait. What do you mean by an allele? Allele represents a particular trait. So what do you know about the trait here? Trait, what is the meaning of a trait? The trait is a specific characteristic of an individual, a specific characteristic of an individual. For example, the person's eye color. How does the person's eye color is? Whether he has got brown eyes or he has got black eyes. Suppose you take a person's texture of the hair, whether they have a curly wavy hair or they have a straight hair. So this is nothing but the trait. When allele is able to carry the information for a particular trait, so we call them as alleles. What do you mean by allele? They are the same genes. Allele means what? They both contain a same gene. They both contain a same gene, but they have a slight difference. But they have what? They have a slight difference. So that is why we call it as a allele, as a variation of a gene. We call them as a variation of a genes. So the way, what will be the variation? They both the genes are carrying a what they carry, what the trait they carry. They either carry a trait for the color of the eyes. So one trait will carry the color for blue eyes, brown eyes. The other one will trait carries for the black eyes. But both of them are the genes coding for the eye color. But they have a slight difference. And when, how do they express? So in the allele, we have again, what type of alleles are present in a human body? They are always two forms. The alleles will always expressed as either a dominant form or in the form of recessive. Sometimes they also express in a multiple alleles. So, but basically here, allele is nothing but a variation of gene. They are coding for the same thing, but they have a slight difference. They can be either expressed as a dominant character or in the form of a recessive character. So next we have a question, what is a genotype and what is a phenotype? What is a genotype? The genetic composition, we call it as a genotype. Whatever the genes present, and that genes we call them as a genotype, and that part we call it as genotype when it is expressed physically, and we call the part as called as a phenotype. What is a genotype? It is a genetical composition, and what is a phenotype? It is a physical expression. For example, when a person is tall, so the genotype will be capital T, capital T. So this is a genotype. When I say the person is tall, this is the phenotype. Again, you think capital T and small t. This is also the phenotype represents this tall. This is also a phenotype and this is the genotype. So what is a genotype? A genetical composition. Phenotype is the physical expression. So what do we understand now? We know there is a typical structure called as a chromosome. Every chromosome has got a locus. The locus is the point where the genes are present. So genes are typically carrying a structure called as what? DNA. The genes are calling they have a DNA molecule in it. Apart from that, the genes also carries a concept called as alleles. What is an allele? Allele is nothing but a variation of a gene. It is nothing but the gene will be the same, but they have a slight difference by expressing either themselves as dominant or a recessive. So what is the genetic composition? We call it as genotype. The physical expression, we call it as a phenotype. So this is about how the chromosome is present, how the alleles work, how do the DNA encodes the information for a particular physiological function. So now we'll go still better way. We'll study about the still more the ultrastructure of the chromosomes by talking about the centromere. So this part we call it as a centromere. The centromere is also has got another name. We call it as a primary constriction. What do you mean by a primary constriction? When both the arms of the chromatids are separated by a center narrow region, we call them as centromere or a primary constriction. So this centromere will have a protein complex. The centromere will have what? A protein complex, and this protein complex will be a disc shaped in structure. We call them as kinetochore. I'll make it very simple. The center region is called what? Centromere. In this centromere, they have some protein, and that protein we call it as what? Kinetochore. What did I say? The center part which separates the chromatid is called centromere. Inside the centromere, you have a protein part, and that protein part we call it as a kinetochore. Now the next question comes. What is the purpose of the kinetochore here? This kinetochore attached to the spindle fibers during cell division. Hopefully you all know mitosis, meiosis. In the process of mitosis, meiosis, this kinetochores are attached to the spindle fibers. during the process of the division. So we were talking about the chromosomal structure. The narrow constricted region, we call it as a centromere. We call this as a primary constriction. Inside this, we have a protein, and that protein is in this shape in both the sides, and that we call it as a kinetochore. The function and the purpose of the kinetochore is to attach to the spindle fibers for the movement of the chromosome during the cell division. So this is about the primary constriction. So this primary constriction is followed by the secondary constriction. So now we come for the secondary constriction. Now we know primary constriction. So now we come for the secondary constriction. So what is secondary constriction? Apart from the centromere, we have an additional thing, we call it as a secondary constriction. Above the secondary constriction, we have a chromosome, and that chromosome we call it as a satellite chromosome. What did I say you? We have a primary. In addition to that, we have another more point. We call that as secondary constriction. This secondary constriction, the part of the body present above the secondary constriction, we call them as a satellite body, or we call them as a SAT chromosomes. These SAT chromosomes contain NOR. This SAT chromosome contain what? NOR. We call this as a nucleolus organizing region. So once again, I'm coming from the primary constriction. When you take a chromosome, the chromosome has got a center part. We call it as centromere. It has got a protein, and that protein we call it as a kinetochore. And that kinetochore helps in the attachment of the spindle fibers during the cell division. This is the thing which we saw about the primary constriction. And now we are going to study about what is secondary constriction. Apart from the primary, they have an additional part, we call it as secondary constriction. Above the secondary constriction, we have a part of a body called as a satellite body or a satellite chromosome. And this satellite chromosome has a special region called as nucleolar organizing region. Now in detail, again, we'll study about what is satellite chromosome, because we'll finish off with the satellite chromosome. These secondary constrictions, what they're saying about this, we are talking again detail about the secondary constriction. They have what region? Nucleolar organizing region. So this organizing regions helps in the, which involves in the formation of the nucleolus. Because of the presence of the satellite chromosome, they helps in the formation of an organelle called nucleolus, and they contain a genes for rRNA. They contain the genes for what? Ribosomal RNA. And this is always species specific. So very, very important point. This NOR is species specific. What do you mean by species specific? Every species is very specific. For example, every organism will have a unique type of NOR. So it is used in the process of karyology or cytotaxonomy. What do you mean by karyology? Where you're going to detailedly study about the structure of the nucleus. We call it as karyology, study of nucleus. Cytotaxonomy. What is cyto? Means cell. Taxonomy means studying in different classification. How could you classify a cell? With the help of this both, you could study the NOR. With the help of NOR, you can have a detailed idea of the karyology and as well as cytotaxonomy. So what we were talking about? We are talking about secondary constriction. When we talk about secondary constriction, primary constriction, additional we have a one more constriction, we call it as secondary constriction. Above that we have a certain body or a chromosome, we call them as SAT chromosome. This SAT chromosomes contain what? NOR. This NOR is involved in formation of what? Nucleolus. This NOR is involved in the formation of what? Nucleolus, and they always are species specific. So that they are used in the technology of karyology and as well as cytotaxonomy. So we will summarize all the points which we have seen in this lecture. So what are the things we saw here? We were talking about chromatin fibers. This chromatin fibers are like, what is this? This is a bundle of wool, and in that we have a stretch, a single unit, we call it as chromatin, and this chromatin on condensation they form chromatid. This chromatid on duplication they form chromosomes. Is it clear? Chromatin reticulum, they have a chromatin, they become a chromatid, they form a homologous sister chromatid, we call them as a chromosome. Once they become a chromosome, they have two constriction. One we call it as primary constriction, the other we call it as secondary constriction. The primary constriction is called what? Centromere, and they have a protein called kinetochore. They helps during the process of cell division. When you talk about the secondary construction, this is called as an additional construction, and there is a presence of a satellite chromosome, and where they have a nucleolar organizing region, which is involved in the process of nucleolus formation. Apart from that, they play a vital role in the study of karyoology and cytotaxonomy. As I summarize the entire concept regarding the chromatin fibers, chromatin is present, chromatid, followed by an X-shaped structure called as homologous sister chromatids. We basically call them as a chromosome. And then we had a clear idea about what is a chromosome contains. So by this, I summarize the entire concept and make sure that you people understand better way. Thank you so much, students.